Hello, my name is Dr. Philip Toleikis, and I'm President and CEO of Sernova Corp. I just want to thank the sponsors for the Gene and Cell Therapy meeting on the Mesa for this invitation. Sernova is a publicly traded company on the Toronto Venture Exchange and also listed on the OTCQB under the symbol SEOVF. We are furthermore listed on the Frankfurt Exchange and are planning on uplisting over a period of time to NASDAQ. Please be aware of the forward-looking statements in this presentation. So Sernova is a clinical stage company developing products for the treatment of chronic debilitating diseases. The company is engaged in the development of its proprietary cell pouch system consisting of an implantable medical device and immune-protected therapeutic cells. We're working on a commercially viable treatment for insulin-dependent diabetes and other metabolic, blood, and rare diseases with therapeutic cells that are placed into the implanted, vascularized, and scalable medical device that we call the cell pouch. With respect to important investment highlights, we have an integrated regenerative medicine therapeutic solution. We have an active phase 2 diabetes clinical trial ongoing at the University of Chicago, for which I will be discussing some of the data that's come out. And we also have preclinical advanced programs using human cells for hemophilia A and thyroid disease. Finally, we have multiple collaborations with pharmaceutical companies for stem cell derived technologies, both for type 1 diabetes and other types of indications that we're looking on at bringing on to clinical applications. So with respect to the approach of the regenerative medicine technology that we're working on, Sernova is really focused on a potential functional cure as opposed to masking disease with drugs and other medications. With respect to our platform technology, there's 3 parts of our technology. First is the cell pouch. And importantly, this is a scalable device that goes deep under the skin, and it basically provides a highly vascularized organ-like environment for the placement of therapeutic cells. And this device is removable and allows those cells to be housed directly into the device. With respect to therapeutic cells, we first work with human donor cells because those are the cells that we know work in your body. But as we're moving forward, we advance with stem cell derived technologies to treat all patients with the disease. We also have worked with therapeutic tissues in our device and found that to be successful. Regarding our immune protection technologies, we have 3 approaches that we're working on. One, for our first clinical trials, are the anti-rejection medications to be able to move rapidly into clinic. And as we're moving forward, we're working on a conformal coating technology that we have licensed in that is a polymer coating. And we're also working on 2nd generation gene editing programs. So in terms of our commercialization approach, we work first with the most critical unmet need patients. And then as we add our technologies in and de-risk our programs, we're able to treat more and more patients. So in other words, as we move away from the anti-rejection medications and the human donor cells into stem cell derived technologies and our local immune protection, then we can treat a larger and larger population. But our anticipation is that we would have earlier products with human donor cells approved first and then followed up by our stem cell derived technologies. So with respect to the cell pouch, we have done a lot of testing, both preclinically and clinically, showing the superiority of our device. We show that the device consistently vascularizes when placed in the body. We show long-term no fibrosis in the device, even after a year of placement in the body. The device is very scalable, and it's very, very versatile in terms of the types of cells that can be used. So we have worked with stem cell derived technologies for type 1 diabetes and shown very, very good efficacy in animal models of type 1 diabetes with our partners. And we've also shown efficacy in humans working with human donor islets, as well as working with thyroid tissue in preclinical models. With respect to the manufacturing, we manufacture our cell pouch in multiple sizes at a US facility under GMP conditions, and it meets the regulatory requirements for the US, Europe, and Canada. In terms of our local immune protection technologies, we have licensed in conformal coating from a company called Converge, as well as from the University of Miami. And there's over 12 years of research behind this technology. And this is a thin biocompatible polymer coating that surrounds the cells, that allows those cells to be protected from immune system attack. And it has been tested both with islets and stem cell derived technologies. So if we look at the rationale for conformal coating encapsulation, whereas traditional microcapsules are quite large relative to the cells that are placed in them, conformal coating surrounds the individual cells and provides for a much better approach in terms of space. It's also been shown that there is no delay in terms of glucose-stimulated insulin secretion when compared to naked islets. And furthermore, with the minimal volumes that these cells take up, it's a perfect situation for placing these immune protected cells into our cell pouch organ-like environment chambers. So with respect to some of the preclinical studies, conformal coating platform has demonstrated both efficacy and safety with primary islets in mice. And you can see on the left-hand side the glucose tolerance test showing that naked islets and conformal coated islets perform virtually the same. We've also shown long-term allograft studies in diabetic mice that we get insulin independence without any immune suppression. And this technology has also been shown to be effective in human and safe in human primates. Furthermore, the technology has been scaled up for larger production of the conformal coated cells. Importantly, as we move forward with our unlimited supply of stem cell derived technologies, it's been shown that conformally coated stem cells also reverse diabetes in mice long term and are able to regulate blood glucose to normal human levels. So with respect to our collaboration that we have recently announced with the University of Miami, our goal is to move as rapidly as possible, combining this technology both in donor islets as well as stem cell derived technologies in our cell pouch, and move this technology into clinical trials as rapidly as we can. We are also working on a gene editing approach with a company called Ajax as a second generation technology. So we have multiple options in our toolbox for protection of our cells against immune system attack. When we talk about our device technologies and cell technologies, we believe we have a biologically compatible delivery process. So what does this mean? What we do is we implant the cell pouch about 30 days prior to placing the therapeutic cells. And during this time, the device fills in its pores with highly vascularized tissue to form the organ-like environment. Then we go in and are able to place those cells into the device chambers, and they're sitting in a tissue matrix that is highly vascularized. So they can rapidly connect up to the blood supply and then start to release their product as required. So our first clinical trials are around type 1 diabetes. And our first indication is really working with brittle diabetic patients that have hypoglycemia unawareness. And hypoglycemia unawareness is a situation where when patients take an insulin injection, their blood sugar levels can drop and they're not aware of that happening. So they can actually go into a coma and potentially die. This is a very serious situation, and we're working with very unstable diabetic patients who have this problem that is not solved even with close regulation of insulin levels. So with respect to our phase 2 clinical trial that is ongoing at the University of Chicago, we have a primary objective as safety and a secondary objective as a number of efficacy measures. And these secondary objectives include survival of endocrine tissue in our sentinel devices that are placed along with the therapeutic device in the body. And our sentinel device is removed at approximately 90 days, and we can assess the survival and viability of the cells that we're placing into the device. We're also looking at the proportion of subjects with a reduction in severe hypoglycemic events, and also with reduction of hemoglobin A1C. And furthermore, we have 20 additional endpoint analyses that we're evaluating. With respect to our phase 1-2 safety profile, Dr. Witowski has presented at a number of conferences, and it's showing that the cell pouch is well tolerated and safe during the implant and time of transplant. And we're seeing no incidences of severe adverse events determined to be probable or high related to the cell pouch. So the conclusion has been and continues to be that our device and therapeutic cells meet the primary endpoint of the study. Furthermore, we have examined the most advanced patients in the study. And in this particular case report that has been presented, if we look on the left-hand side, you can see that prior to islet transplant, this patient is having 4 severe weekly hypoglycemic unaware events. You can also see the amount of insulin they're being used and hemoglobin A1C. With the Sernova cell pouch transplants, we're showing a reduction in hemoglobin A1C, a reduction in about 50% in the daily use of insulin, and also, very importantly, a reduction to 0 in the severe hypoglycemic unaware events. So even with this reduction, we're seeing that this is a potential product. Furthermore, we're consistently showing blood levels of C-peptide, which is a biomarker for production of insulin by our cells in the device. After we placed a top-up of cells on the right-hand side, you can see that the hemoglobin A1C dropped down to 4.9, which are normal levels. The patient then went to 0 need for daily use of insulin, and as long-acting and short-acting insulin, and continued with the 0 hypoglycemic unaware events. So this is what we call a functional cure. Importantly, this patient has provided us a short testimonial. And after 47 years of insulin injections of being diabetic, this patient has been off of insulin for over 18 months now. So we look at this as a sustained effect and something that we would like to achieve in all of our patients going forward. So what's going on with the rest of the study? So the study has 6 patients enrolled, implanted with cell pouches and ongoing islet transplant. We're continuing to show safety. And from an efficacy perspective, Sernova, we believe, is the first regenerative medicine company to demonstrate a subcutaneous islet transplanted device achieving persistent islet graft function in type 1 diabetic patients. We're seeing clinical benefit reported by the investigator in case studies in our patients, including reduction of C-peptide or sustained levels of C-peptide, reduction of HbA1c, improvement of continuous glucose monitoring, and reduction in the need for exogenous insulin. And importantly, the investigator has shown that even a reduction of the number of cells in the device improves the efficacy. So in terms of our commercialization strategy, our goal is to complete the study, and we're looking at next steps in terms of pivotal study pending FDA clearance. And importantly, we're looking at working with patients in terms of our conformal coating technology to eliminate the need for the anti-rejection medications, both with human donor islets as well as with stem cell derived technologies. So with respect to our hemophilia A program, I just want to say that we have already shown with human donor cells, with human cells that have been corrected for the gene for factor VIII. So this is an ex vivo gene therapy program whereby we have been able to, in preclinical models, correct the disorder, the bleeding disorder in these animals. So this is something that will be moving on into clinical trials. Furthermore, we believe we're the first company in the world to be able to be working with patients that have thyroid disease, and these patients are having their thyroid gland removed. And what we're looking at is approximately 150,000 thyroidectomies per year. And Sernova's approach is actually to take some of the healthy thyroid tissue that is remaining and then place that into the cell pouch. And we've done preclinical studies showing already that this works. Our goal is to be into the clinic with this program in 2022. With respect to our patent portfolio, we have an international patent portfolio around medical devices and transplantation, the stem cell derived technologies, and our local immune protection technologies. And with respect to our upcoming milestones, as I mentioned before, we're working, moving our type 1 diabetes program forward, our local immune protection technology forward, and then with our human donor islets, and this will be followed up by stem cell derived technologies. We're also planning on moving our thyroid program into the clinic in 2022. And from a corporate partnering perspective, as I mentioned earlier, we're working with multiple pharmaceutical companies and looking at getting licensing deals done with stem cell derived technologies. So we have, in terms of our corporate team, very experienced executives, as well as a very, very well-seated board of directors. And in terms of our share structure, we have a strong cash position of approximately $32 million and a market cap of $334 million currently. And for more information, please note this slide.